By Dan Wade here from the University of Hull, who will be discussing modelling avian influenza in the UK, an ecologically targeted approach to risk management. So without any further delay, over to you, Dan.
Thank you, Victoria. And hello, everyone. Thank you very much for inviting me to lunch talk today, Victoria. I'm looking forward to going through what is essentially my PhD thesis. So I am late stage in the game with my PhD at the moment. I'm very nearly ready to press that submit button. But this is actually a fantastic bit of practice to help me make sure I've got all the narrative right in my head and to kind of give off some of the results that haven't really seen the light of day yet. So hot off the press. So if you give me a moment, I will share my presentation with you. So yeah, you know my name by now, and my talk is modelling avian influenza in the UK, an ecologically targeted approach to risk management. So I wanted to really begin with a bit of background and kind of introduction to how the project began its first iterations in life. So we're talking back down in 2017, 2018, when actually the avian influenza landscape in the UK was quite different to what we're finding it today. And back then, the kind of main focus of motivation for research into avian influenza took a poultry farm focus. So we know that outbreaks do occur in poultry farms in the UK, and we know that the impacts of them are mostly economic, and yeah, there's a lot of birds have to be killed off in outbreaks. So is there a way that risk can be reduced to these poultry farms? And the thesis is kind of structured with that in mind, and also looking at what variables could be involved in avian influenza outbreaks in the UK, thinking about wild birds, thinking about movements within that and their ecology, and can we monitor AI in wild birds a little better than we do today? So a little background for avian influenza. So you'll notice I write AI a lot here. I'm not meaning artificial intelligence. I'm talking about avian influenza when I do that. So whilst we've known about avian influenza since the 1950s, our understanding has been limited by the ecology of their wild reservoir species, which is wild ducks. So a lot of the research that is done on wild birds focuses a lot at ducks, geese, and swans. And then behind that, it's mostly water body centric families. So we're talking gulls, waders, and stuff like that. So the terminology and understanding of symptoms within wild birds tends to be defined by the effects of an avian influenza strain on poultry. So you see HPAI and LPAI, that's high pathogenicity avian influenza and low pathogenicity avian influenza. So high path and low path, if we will. High path tends to be high mortality and morbidity. Low path can be as little as a cough or a sneeze, and even can be asymptomatic in poultry. But those definitions don't necessarily stand the same in a different host bird. So in a duck, a HPAI may very well actually not affect the duck too much. So furthermore, in the background, I'm not going to go too much into the kind of makeup of the virus itself, but to understand the complexity of the different strains of avian influenza, the virus is made up of kind of two units, the hemagglutinin and the neuraminidase, and that they have essentially lots of different types, which are numbered, so H1 to H15, N1 to N9. And in terms of the concerns to do with avian influenza, a lot of it is to do with how avian influenza might find its way into the human kind of chain of life. And there historically have been strains found in humans, which the kind of main concern one is H5N1. So there was quite a sizable outbreak of H5N1 in the early 2000s, around 2003, which had a pretty high death rate in humans. We're talking kind of 50%, and that tends to ring big alarm bells. So things have died down a bit since then. There isn't this kind of large scale outbreak ongoing in people at all, but things have changed in the landscape for AI. So I've mentioned already about the kind of in poultry, when AI gets into the system and it's detected, you kind of have to kill off the flock. So that's big economic strain, and actually the resources associated with that is yeah, it's quite a lot. There's a lot of money goes into monitoring, a lot of money goes into management. But if you think about the kind of the UK supply chain and the amount of protein that is provided for our tables through the poultry industry, it's quite a lot of the chunks. So it's an asset to be protected. Monitoring of avian influenza in wild birds is a little more tricky. Generally speaking, the historic way of monitoring for bird flu has been through finding dead birds in the landscape, reporting them in. The Animal and Plant Health Agency, the APHA, will send out a team to collect those birds, and then they will be tested. So it's quite it's a resource intensive way of monitoring. So bringing it kind of forward a bit, I've mentioned the motivation from a few years ago, but today we know that avian influenza has really kicked off within the UK and worldwide. Since in the last couple of years, we have seen everyone likes using the word unprecedented, but it is. There's been so much AI in the landscape, wild birds and through poultry outbreaks. These are the biggest outbreaks that have been seen in the UK. And that's, it's causing a big problem for supply chain, and also thinking about preference for free range goods. If you have to lock away your poultry to mitigate outbreaks, you will eventually lose that status. So there's lots of things like that to consider. So as this is increasing and that focus has come round, it has helped define the directions the project's taken. So can this project help understand the AI landscape in the UK? I hope so. So now getting on to the thesis itself. So I had 4 research chapters. My first chapter kind of looked at it from a very broad systematic literature review, a lot of reading, a lot of number punching, essentially, of AI sampling in wild birds. And then I moved on to look at sampling for AI, which took the form of a now published paper, which is titled High Pathogenicity Avian Influenza, Targeted Active Surveillance of Wild Birds to Enable Early Detection of Emerging Disease Threats. And then chapters 3 and 4 take a modelling approach. So the first is looking at modelling around poultry farms, which is our chapter 3, and who's present there and what risk they might bring. And chapter 4 uses eBird data to look at co-occurrence of these species that are present at poultry farm at water bodies as another identified site of risk. So moving on to my first chapter of my thesis, this is the systematic literature review of AI sampling in wild birds. It was a lot, as I've said, of reading and a lot of taking data. So original kind of very broad search term of avian influenza brought out 1,314 different article titles. And when you scan all that through various different processes, which I won't go into too much detail because it's a little bit boring, that we get down to 116 data extractions from different articles. So 116 different articles provide 893,993 samples, which, that's a lot of lines in Excel, I can tell you that much. That's about 371,007, which were tested for IAV being also being AI, another symbolism for that. That's viral presence, and then 25,000 and a bit for presence of antibodies, which are seroprevalence. The motivation for doing this is, can we identify areas for further research from patterns emerging from current sampling data? And does the sampling bias match the AI prevalence in different species? And can these findings help structure the rest of the thesis? That was the motivation for doing this. So once you've got all those lines in Excel, you can align each sample to be different to its species, and we can start looking at who is sampled the most and what proportion of positivity is there, and also looking at the certainty in that kind of example. So I've actually just picked the top 20 species from that table. There is over 1,000 species in this data set. So what I just kind of want to point out here is, if you don't know your binomials, most of these are ducks. Ducks are way more sampled than anything else when it comes to AI wild bird sampling. Obviously, there's a lot of unidentified samples there on row 3, and apart those that aren't ducks on that list, there is coot on there, cattle egret, but they are and tend to be water body centric species that hang around at water body habitats. So, kind of taking this to the next step is looking at how these families kind of fit together. These species kind of fit together, their behaviours, and what that might mean for the avian influenza landscape. So this next graph is kind of the results of 2 models. I think both GLMs looking at the kind of different spread of species and their prevalence rate and their sampling bias for different metrics. So kind of key important ones here is that wetland-centric species are sampled more, and they also have a higher proportion of samples that come back as positive, and they both are significant. So if you're just for reference, if a line crosses this 0, it means there's an uncertainty of direction, which essentially means it's not a significant p-value. So yeah, there is uncertainty in the kind of colonial nesters here for the sampling bias, and also for oceanic species, which I added in after the big scale outbreaks in seabirds we've seen in the UK over the last couple of years. So yeah, okay, it looks like there's a trend there. Interesting. Oh, I've stopped moving. There we go. So now highlighting this towards this is a graph that I don't expect you to be able to read all the things on here, but this is the Anatidae family and all the species that have been sampled within it. What I want to highlight is that it's easy to look at a graph and say, oh, there's a lot of ducks involved here, but actually the uncertainty within a family is quite high. So the more you sample, the less your uncertainty tends to be. But you can see by this landscape that there is a massive degree of like, do we really know within Anatidae if there's one species? Like, are mallards really key in this? It's just that there is a lot more confusion when you dig into the data. And just another example here is I've taken out another family, which is Acrocephalidae, which is the reed warblers. So they are another wetland-centric species for the most part, or families for the most part. And that there's still high levels of uncertainty and much smaller sample sizes, but that spread of data is still evident. There is a lot of uncertainty in what we can say about that through current sampling. So what I'm thinking about this and thinking about this influence that Anatidae might have on that data set, I decided to run things through a kind of all species and a non-Anatidae species model approach. So in terms of sampling bias, it exists for all species, and when you remove Anatidae from the testing, it's still it's still there. It's still higher, a less degree of severity. We look at the proportion of positive samples. There is a degree of overlap with all species, but that degree of overlap increases a lot for kind of when we remove Anatidae from that model. So actually, that trend towards wetland centric that we saw in the graph a couple of slides ago, possibly not quite that all encompassing story here. So running the same model again without having Anatidae in, actually we see everything is crossing the 0 line, so there isn't significance in wetland centric species for sample size or proportion positive. So that certainty on wetland centric species being key players outside of Anatidae is uncertain. It certainly leans one way, but it's uncertain. So digging this back again to look at things from a family level. So this graph here is looking at the kind of percent positive of a species within a family. So for Anatidae, again, it stands out quite a lot here. We have Ardeidae, which is the herons and egrets, Laridae, which are the gulls, stand out. But we have some outliers out here, which is Alcidae. At the bottom there's auks, and Scolopacidae, which are waders, sandpipers and allies. So the reason I put this up is to kind of demonstrate that this is taking all the data above species that have been sampled over a sampling threshold, which was calculated through epidemiological modeling calculator, which is 738. So by doing that, we indicate that there is a certainty that you would have detected a positive sample if you've sampled that much. So there's obviously a swing in bias towards wetland families in this, so they are going to be the most representative. But let's just kind of take a step back here and say, okay, what if we take a sampling threshold of a bit lower, so 300 and above? So that was something done in 2 other systematic reviews that I've referred quite heavily to in my chapter. So that actually changes the scene quite a lot. When you take more species in, whilst there will inherently be more uncertainty, we certainly have other families starting to feature here. So that kind of high blue in the middle there is Passeridae, which is sparrows. At the base there, we have Acrocephalidae, which I've already mentioned, the reed warblers. And you can just see it's a little bit more uncertain. And then if you do that again, just to prove the point, that's 100 and above. And things just aren't quite as clear that Anatidae are the big player here. So there is more research that needs to be done to figure out the role of other species within this landscape. So to summarize this chapter, AI has been found among many families and species if you look hard enough to find it. So yeah, research effort could go elsewhere and we could find more data. Taking an approach of anyone could be infected may identify some gaps in our understanding of AI prevalence and spread. Whilst research focusing on state is unwarranted it needs to be understood that it can't provide all the answers we need. And lovely, right here I've noticed I put wetland bit because I wanted to talk about wetlands and apparently I didn't actually write anything. So the wetland bit is that you know, we're not 100% certain that wetland centric species do have a higher rate of AI than non-wetland. What we do know is it does seem that ducks influence that quite heavily. So ducks seem to be where we need to look, which is something that has been identified quite heavily previously in the literature. The kind of key points to take away from this is kind of the who. If you took it from a UK context, there's a high level research in Anatidae, which, you know, it seems to be warranted. With further understanding developed around species found dead for monitoring purposes, which tends to be larger species that are easier to find. So systematic review suggests that there are other pieces of the puzzle to connect to build a fuller picture. So we need to know the roles of other species here. Dataset for AI prevalence per species from systematic review, plus errors, make assumptions, but is a useful indicator. So, you know, these are broad strokes. There is a lot of nuance to what defines a prevalence rate. This is very much taken on a population level, but it's a good indicator of AI spread among species, especially for those species that have been sampled more than a few times. But we do need more sampling of wild birds to understand the roles of less sampled species and their spread. Okay, chapter 1. Now we're on to chapter 2. So this is the one that I have published, which is, you know, very excited about. It's my first author paper out there. So the title is High Pathogenicity Avian Influenza in Targeted Active Surveillance of Wild Birds to Enable Early Detection of Emerging Disease Threats. What does that mean? Well, it all starts in Yorkshire. So my PhD was heavily linked with Spurn Bird Observatory, which is the bottom right-hand square. And I also did some sampling of wild birds during mist netting at Filey Bird Observatory at the northern square. And then this kind of third square is about a different approach that we take, which I'll go on to next. So we trialed 3 active sampling strategies to try and monitor for avian influenza in wild birds during autumn migration. So there at bird observatories like Spurn and Filey, there is kind of systematic sampling or systematic bird ringing that goes on at those bird observatories throughout the autumn period. So I took the opportunity to take fecal samples during that autumn migration window, which were then tested for avian influenza. Our second kind of method was sampling passerines at locations around outbreak sites. So in early 2020, there was an outbreak at a site in Suffolk, and I went out with Adam, who was one of my supervisors, and we went and caught birds at different locations in the area to see if we could find AI in that population. And then third was sampling from hunter-harvested waterfowl during autumn migration. So that's a bit of a different approach. So it was good fun. Put some photos in. There was a lot of screwing bits of bird poo into little tubes. I got very few different nicknames of my time at Spurn, as Yorkshire does provide. And yeah, but a lot of frosty early mornings catching birds on migration, which, you know, that's something is very much that I've always enjoyed. So yeah, that was a good part of the thesis for me. And caught some lovely birds at the same time. So any bird watchers here, yeah, the two on the left are actually quite uncommon that we got sampled. It's a red-flanked bluetail at the top on the left. Bottom left is a yellow-browed warbler. Top right is one of my favourites, which is a brambling, and then the bottom other one is red-legged partridge, which you can't actually ring in the UK, but I did get a sample from after it decided to bumble into a mist net while kind of ringing at a farmland site. All very nice species to see, and yeah, very enjoyable. The results came back kind of, yeah, with different mixed bag really. So no avian influenza was found in migratory passerines and their allies during autumn migration sampling. So the little guys that are moving did not return any positive samples. And there was no avian influenza found during responsive sampling of passerines locally to an outbreak site in Suffolk as well. But the hunter harvested waterfowl did produce positive samples. So 7 ducks came back as positive. So of the 7 different species that were sampled, there was 7 different ducks of 3 species that came back as positive. So we have teal, widgeon, and mallard. So the samples were tested for avian influenza presence. So that's 7 positives there. There was 3 of the birds tested positive for H5, which you could be H5N1 or a H5 strain, and then 2 of those 3 tested positive for highly pathogenic H5. So the kind of the big scary one, so to speak. So during the hunting, fortunately, but it's very difficult ground as an ecologist to know what to refer to all this. But it's the teal that were shot during the practice. Three did come back with rings, and we have kind of recovery direction. So you know, it generally follows the trend of bird migration in teal as we understand it. We get an idea of where these birds are coming from when they're going into the country. So these birds were shot on the same days as birds that came back positive for avian influenza. And then this leads me on to this graph, which looks at how these birds are moving into the country and when and why that's important. So the sampling that we did actually began in mid-October, and we got positives for HPAI pretty quickly in that active sampling window. But that doesn't necessarily match with when the birds are first coming into the country. It's probably around the peak time for migration in teal and widgeon. But the migration can start as early as August and tends to really start to ramp up in September. So whilst we started our sampling then, as that was the earliest opportunity to do so, you know, there needs further testing to see what happens in that window previously. And kind of one of the big findings that we had is that our active sampling method had detected avian influenza quicker than the passive monitoring system that the government was using at the time. So that's for the whole UK. So using one site, we'd kind of beaten them to the punch, which is, you know, very interesting, and you know, could present some opportunities going forward for different methodologies. So yeah, to summarize, using citizen science and hobbyist practice, it's possible to survey for AI in a cost-effective way and more efficiently than through a passive method. While sample size is too small to be conclusive, results suggests, hang on, block my screen, it suggests that AI is likely to be moved into the country with migrant nattidae rather than passerines. So it's a difficult one to say that passerines don't move it, because you know, you need quite large sample sizes to be able to say definitively, which is one of the reasons why it's difficult to take so much from the systematic review. But the indication is that nattidae seem to be where to look. And then, yeah, it's points to take away. AI prevalence in waterfowl during autumn migration is detectable through an active sampling methodology. And since this work kind of happened last year, year before, They have repeated the method through the APHA, and my supervisor, Dr. Alistair Ward at the University of Leeds, they have been repeating this throughout last autumn. I know they have results that I can't talk about, but I can say that there are more positives that have come out of it. So the kind of proof of practice in 2 separate windows, and yeah, other species of Anatidae were detected with positives. So it's kind of a watch this space for when those announcements come out officially. And whilst passerine roles within immigration movement of AI could be minimal, as I've really mentioned, their further dynamics, the further role in AI dynamics could be different elsewhere in the viral landscape. So now on to third chapter. Hope I'm keeping you all here. And this is about modelling. This is using count data from poultry farms in Yorkshire, and kind of looking at different values for their presence and their absence, and also looking at epidemiological values about, I'll kind of dig into that a little bit as we go on. First, I want to try and explain to you the kind of a mind map almost of what it means to have this kind of movement around the landscape. So poultry farms obviously our central focus here, but there are other identified areas where birds tend to congregate, and others that are less obvious, which I've just labelled as other ecotopes. But the idea here is that in each of these landscapes, there is direct and indirect exposure of avian influenza. The direct is a species to species exposure. So species 1 coughs on another species or individual and spreads it that way, or defecates and one dabbles it up. Ducks aren't particularly clean creatures. So yeah, in the indirect is kind of environment to individual exposure. So if you're thinking of a kind of a duck defecating into a water body, that could then infect the water, that could then be dabbled in by other species, and that could spread through that. Or another example, so this is called fomite, when you kind of leave a deposit of material, be that a cough or a poo, essentially. And then a bird might tread in that and then fly to an important area, such as a poultry farm, and then deposit it in that area, and then it can move that way. It's extremely complicated on how this could work, and it makes studying it actually a nightmare, really. There are lots of avenues to explore, but the key ones that I'm looking at here is that we have species communities that exist at each of these sites. Are there species that exist in these different communities at the same time? Are there birds moving between these communities, and what does that mean? So this wonderful piece of artwork that I've put together here is kind of looking at how I looked at modelling avian influenza at poultry farms. So I went along and did the counts, but I was counting birds that were within what's considered biosecurity fencing. So that's mostly defined by their kind of aspect on people. It's like people need to do certain things to enter these premises. But birds don't. Birds can fly in. It's still a risk area. It's the closest area to the poultry sheds, and it is, it defines probably the highest risk. So there are other birds that are utilising the landscape surrounding outside the biosecurity fences. So my counts kind of went out to about 50 metres outside of these fences. So I've considered that and called it landscape risk for the species present there. And there are also species that fly over these poultry sheds, and what risk do they pose when they could be defecating over these sheds and producing matter that could infect. So risk is probably more minimal on that side, but it's still important in the further landscape. So model construction. So as I've identified 3 areas, each of these areas has 2 separate models. So there is what I've called a general exposure risk score. So this is how many birds are present and how often they're there. So I've used Monte Carlo simulations to produce these data sets. So it's I won't go into too much detail on that methodology. And then using essentially AI influenced exposure risk score. So that is kind of taking that kind of first model and then adding a bit. So we've taken the kind of size of viral load, so essentially how much they're shedding, or it's very difficult to get accurate data on that, But a good proxy for how much material is being left by an animal is to look at their body masses. So I took measures from Dunning et al. 2008 to do that, and then the prevalence data from my first chapter was used to look at the prevalence rate of AI within these species and how that might affect risk and affect understanding for these sites. So going forward, this is the first graph of 6 that look quite similar, so I'll explain these. So this graph is the first thing to note is that each of these are BTO codes for different species on the base, and the scores are relative to one another. So ones that stand out highest are going to be the ones to kind of take note of. So these birds are birds that were found most abundantly and most frequently within the biosecurity fencing. So the big standout there is pied wagtail in the middle. They were definitely by far the most prevalent species around biosecurity fenced areas. And yeah, that's interesting in many different ways, looking at behavior. There's tree sparrow also stick out a little bit here, and then yellowhammer have a bit more uncertainty on the right, but were present as well. So yeah, there are others kind of just because they are low presence or low prevalence in this graph doesn't actually rule them out. They're still in one of the more high risk areas. So each of these species is important. So just for clarity, there's blackbird, blue tits, carrion crow, dunnock, kind of all in there. Red-legged partridge, tree sparrow, wood pigeon, great tit, wren, and yellowhammer. So now, if we take this same area, so this is still the biosecurity risk, and now I've added in the avian influenza kind of risk metrics. So you can see here that red-legged partridge stick out really quite a lot here, but it shows a higher it stands out as one of the higher risk, but also there's a lot of uncertainty going on here. And that uncertainty is kind of key across a lot of these species in that actually it is very much uncertain if one stands out above the other when you look at our current understanding on the avian influenza levels in these species. So yeah, at this time, uncertainty is kind of stemmed from our understanding of avian influenza prevalence in the species that are present at poultry farm sites. So next graph. This one here looks at the landscape part of the model, so who is present in the area, which is this here again is back to abundance and frequency of species in these areas. So this is without the AI data, wood pigeons stand out quite a lot on the right there, as do starling, tree sparrow, pied wagtail, redwings also in there. But essentially, there's certain species that stand out more than others in this, and the importance here is less so than those in the biosecurity fencing, but still probably more important than flyover, though it's very difficult to quantify exactly what those levels are. So next point here is, again, this is the same model, adding in the avian influenza side of things. Again, it's the same story. There is a lot of uncertainty stemming from essentially putting this AI data in there. It's making the picture a lot more unclear. So and then finally going into the flyover models. So here we have again got wood pigeon, starling, and actually herring gull pick up quite a bit here. So that is kind of the first real indication of a wetland centric species appearing as flyover in these landscapes. And another thing to point out here is that there is also hooper swan, mute swan, mallard, grey-lag goose, cormorant all flew over during these surveys. So this is the only time we're really picking up on species that inhabit water bodies, and Anatidae kind of family species. They're not present on the ground around poultry farms. And yeah, again, this is adding in the AI data for that flyover. It creates a lot of uncertainty. And what I do want to highlight here is on that far right, there's the kind of pink box that stands out. That's hooper swan. So the size of hooper swan and mass is large. The avian influenza rate is actually quite little understood for an Anatidae member species. This actually represents a single flyover of 37 individuals during kind of the kind of March period where they're starting to move around a bit. So the question is herring gull on the left here, which actually has a bit more certainty than some of the others. Is that event where herring gulls fly over regularly more important than a one fly over run of Hooper swan? That's the kind of things and questions that we can ask out of this data set, and yeah, or can kind of question which way it could go either way. And yeah, it's important that we gain a better understanding of this. So to summarize this, yeah, risk modeling is difficult with understanding or current understanding of AI prevalence in species prevalent at poultry farms, creating a lot of uncertainty. Looking at the frequency of species occurrences at poultry farms shows standout species to consider. So I've mentioned pied wagtail within that. That's probably my big standout from that group. Whilst the difference between the three models is unquantifiable, it is most likely that species within the biosecurity fence pose the greatest risk of AI spread to poultry. So you've got to remember that species that are seen within this fence aren't necessarily species that are going inside the poultry sheds, because we're talking about housed poultry for these surveys. So this is birds that are depositing fomite as closely to these sheds as possible. It might then be that something else is getting that material into a shed. And then wetland-centric species are most present as flyovers, providing only small fomite deposition risk to poultry farm landscapes. And yeah, the kind of takeaway points are the role of AI spread in a species is different across landscapes. Immigration through waterfowl is proven, but they are not prevalent at poultry holdings. Passerines are likely to play a role in spread to poultry, though movement of fomite into or near sheds. So through movement of fomite into or near to sheds. It's important to consider other routes through rodents and other vectors. So I've mentioned here that fomite could be deposited. Perhaps a mouse or a rat could pick something up and move it. We don't know. Do we know if a rodent could get avian influenza? How would we know that? There's questions that are still available to be answered. And then this kind of final chapter, and realizing my time is running quite closely here, I will speed through this a little bit. So I've used eBird to model AI interest species co-occurrence at water bodies. So this is species that we've identified through the previous chapter, those that are closest to poultry farm sheds, interacting with water body sites. So I've looked at anatidae within this, as we have such a great understanding of, or a greater understanding of avian influenza prevalence and rates within anatidae. They're kind of seen as the most important group, really. So if we know how often a pied wagtail is present at the same time as mallard, we can start to get a bit more of an understanding. What we don't know from this is if those birds then fly directly to a poultry farm. Just because they're present in both landscapes doesn't mean an individual is present in both landscapes, but that is an extremely difficult thing to figure out. So getting the caveats in early there. So this graph here, this looks at this kind of co-occurrence data of each of our interest species from the poultry farms with 18 different species of anatidae that were present on the eBird counts. So who is present at the same time and who really sticks out? The answer is that kind of, in terms of sticking out, there's a lot of overlap. It's more those that don't stick out here that really pick off. So got, yeah, barn owl, little owl, yellowhammer, and I think that's red leg partridge in the centre. So yeah, we're figuring out who isn't there, but a lot of common species are present in both landscapes. Then I keep calling it a butterfly plot, which isn't strictly true, but I quite like it. So this kind of shows how that graph looks when you kind of take it into the individual anatidae species. So this kind of can point out who at water bodies has interacted the most with poultry farm risk species. So our highest ones there, I don't expect you to be able to identify all the colours, but there is mallard and teal, the top two, with widgeon kind of popping in there as well. So yeah, they're the real key interests. I think grey leg goose also comes in. So I am finishing off this chapter at the moment. This is the kind of the end part for me. So I do plan to look at it from a general presence of poultry farm risk species at water bodies, so not just their interaction with other species in those habitats. And then to factor in the AI prevalence of anatidae, so as we know a bit more about the prevalence and the kind of level of error in that is less. It's more certain. There's more runs and there's less uncertainty. So that will help us gain that better understanding, hopefully. But yeah, I won't present that yet because it's not quite finished. And then I just want to get into these key takeaways from the PhD thesis for me is that avian influenza infects more than just ducks and attidae, and the role of other wild birds play in the UK AI landscape is uncertain. At poultry farms, wild birds present tend not to be water birds, which are tested most for AI prevalence. However, birds at poultry farms tend to be common species which can also be found at water bodies. So monitoring could be improved by looking at other networks that interact with waterfowl, i.e. bird ringers and waterfowlers. That could present more cost-effective routes than we currently use. Future research needs to further sample for AI and common species to help reduce uncertainty in risk models at poultry farms and for many other plethora of reasons. And we don't have all the pieces to the puzzle. How do birds move between key landscapes and how frequently? Is one flyer event of a high AI prevalence species more important than continuous exposure from a more common species? At this point, we don't know, but we can highlight that these events both occur. And that is my end point, with a big thank you to all of the organisations listed here who have helped me at one stage or another of my PhD. I hope that wasn't too boring for you all. A lot of talking from me, and I'll pass the floor over now. Thank you very much.
Thank you for that. That was really interesting. So we've got some questions which I'm gonna ask you.
Well,
so does your thesis consider the prevalent use of antibiotics in farm poultry and the vulnerability to antibiotics resistant diseases / AI? What does this mean for wild bird populations?
I'm afraid my thesis doesn't really look at the kind of antibiotics and the poultry side of things. So whilst it is poultry focused in one regard, we're more looking at the wild birds and how they are interacting with that landscape. So I haven't really factored that in at this point. So sorry, I can't really answer that one.
Thank you. So what is the current status of AI in migratory species across flyways? I recall there was some concern about spread from the UK into the Arctic and then across into North America.
Okay, so the current scene for AI in wild birds and through the flyways it's not a pretty read, to be honest. It is everywhere. It really has, the outbreaks that we saw in our breeding birds in the summer. That's something that's unusual. It has happened around the world in previous places and different locations, but those birds that are now congregating in their kind of wintering grounds or the kind of other breeding grounds in the south for other species, them colonies are now having die-offs of avian influenza. You can't stop birds moving, and a bird is going to move as a bird moves. So it's interesting, the kind of narrative that is spreading to the Arctic, it's often displayed the other way. It's actually birds breeding the Arctic spread the disease amongst themselves and then bring it on their winter migratory routes and then spread it from there. So it's all a big web of connectivity. And this time round, it is literally everywhere. There's big events happening in South America at the moment, which is about as far away from here in the UK as we can get. But there will be links in how this works. It tends to be the different strains evolve in different areas of the world and spread kind of that way. It's when two different strains can come together, can cause a bit of problem and worry. But there is a lot of it, unfortunately, and it's very difficult to manage from a human perspective.
Yeah, well, it's complex, isn't it? So we've got two questions, but I think it's similar. So zero prevalence in passerines could just indicate that infected birds die rapidly, and then also they don't fly into mist nets. Or do you think they genuinely weren't infected as much?
No, these are good points because it's not as simple as that. There is certainly different caveats involved, and yeah, it could be that birds are dying if a house sparrow, and not a migratory species, so poor example, but if that did get avian influenza, if it died, firstly, would we know about it? It's a very small bird. Would it be eaten quite quickly? We have, I know that we've had in the UK foxes found with avian influenza and otters as well. They could be scavenging small birds that we just wouldn't detect. So there's things like this. They might not find their way into a mist net, or we just know that within a nattidae, it does seem that they can spread fairly asymptomatically to an extent, but there are certain species that do die off. We've seen die-offs in barnacle geese and pink-footed geese, so that is an example within family. So it just varies. It's why it's so complicated to study. Each different species is going to have a different outcome. But yeah, it is something discussed kind of in the paper, is that there are different alternatives to the narrative.
Okay, so yeah, read the paper.
Yeah, sorry.
So testing in ducks was just detecting live virus and suggestive of 20% incidence of live virus, so 7 out of 146, of which H5 was in 1.3% of birds, so 2 out of 146.
Yeah, something about that. Yeah, that the numbers rock up. So yeah, that was the numbers we were catching. So this was on the Humber Estuary, this site. So this is just one site that with one waterfowler collecting samples for me over it was October to January. So yeah, I know that this year that they've spread out this kind of sampling effort for different sites. So there are multiple sites around the country. So the results that come back from that are a little bit more spatially spread out. But yeah, I mean those numbers are correct as far as I can tell. Or I can't automatically do the proportion math off the top of my head, but yes.
So was it live virus? Yeah.
Oh, sorry. Yes, it will have been live virus. Yeah.
Thank you. So we got another 2, unless people then want to ask more questions. So how much were humans considered for transportation, particularly of fomites via feces, or is it biosecurity prior to an outbreak always good? For example, washing boots, etc.
So the sites that I was at, they were all for the same company, and they had pretty good human-based biosecurity metrics at or measures, should I say. Everyone was wearing hazmat suits. Everyone had to do dips, and they all had to shower before they went into the premises. There was foot dips between sheds, and it tends to be that in the more Western countries, to be very generalistic, their biosecurity measures for people are quite a bit more advanced than elsewhere in the world. So it's deemed that the risk from that, as long as people are following the measures that are put in place, is probably quite low. So yeah, to answer that question.
Thank you. So how reliable is fecal sampling for determining AI prevalence? Could this approach fail to detect infected birds?
It's never going to be 100% on every different method, just to say there's always levels of error, but there are papers suggesting that it's comparable between cloacal sampling and fecal sampling. So yeah, there is a degree of error, but it's comparable to other methods. So one thing to say that within the UK, if you want to do cloacal sampling or tracheal sampling on, or even blood sampling on wild birds, you need to get special licensing, and there's quite a few hurdles to kind of get through to do that. That was not something we had timescale to do as part of the PhD project, else it would have been something I would have explored.
I mean, if it's comparable, probably it's easier. So.
And that.
Yeah.
Sometimes you go for the easy option.
Yeah, exactly. So did you get blood samples to look at antibodies? This would allow us to detect past exposure and help address the question of infected birds might not fly into the nets.
Yeah, so yeah, this is again, it kind of follows on from the last point in the fact we didn't. I would love to, because I think that is a key part of the puzzle. Seroprevalence is the kind of word tends to be used. Finding out who's had what when is really important. So one thing, or what kind of, there's a paper that I've read that looks at juvenile naivety, it's called, to virus exposure. So often young birds can catch it better because they've not been exposed to the antibodies previously. So if there's a good breeding season, that could change the dynamics of spread. There's so many micro caveats in everything. It's an extremely complicated virus. All viruses are complicated, but in a wild species of animal, it's really difficult to exactly pin down these things. But I think that would have been a really nice piece to add on to my thesis if it had been able to.
I think you probably can have like several PhDs on this topic.
Oh yeah, it's crazy how much new literature comes out every week. It's a lot to keep on top of.
Definitely, thank you. So yeah, these were all the questions on the Jamboard. If anyone has any more questions and they want to ask directly. Okay, so that was it. Thank you very much. It was really interesting, and yeah, I enjoyed it. So
My pleasure. Thank you all for listening.
And yeah, good luck for the rest of
Thank
the
you.
PhD and submission.
Yes, only a few more sleepless nights left.
Thank you.
Thank you very much.
See you, everyone.